Oh hey, we didn't see you there. So today we'll be learning about protein synthesis.
Proteins control all reactions, give structure to organisms, defend and transport fundamental molecules. The double helix of DNA is unwinded by RNA polymerase, which plants itself on DNA a specific sequence of bases called promoters. In transcription, various strands of RNA remove themselves out of the cell nucleus into the cytoplasm.
I'm sorry, cell nucleus, but I have to remove myself from you.
Well, we have so much together.
I'm sorry. Let's go, cytoplasm.
Transcription from the mRNA from DNA, the DNA genes into the nucleus.
Go.
I think that's why it is
Within the nucleus, DNA needs to travel across the ribosome by being guided by the mRNA. RNA polymerase moves down, unwinding and splitting the DNA. What is actually splitting up the double helix is the RNA nucleotides. Double helix unwinds, the bases connect themselves to the DNA template strand. In DNA replication, the RNA polymerase only assembles in a 5' to 3' direction. The difference between DNA and RNA is that RNA uses uracil instead of thymine. The RNA polymerase will eventually stop transcribing when instructed. From then on, the RNA will be released and become mRNA.
Stop.
The next stage calls for 5' cap and a poly A tail. These help protect the mRNA, which guides the mRNA out of the nucleus, and attach the mRNA to the ribosomes. Ribosomes are made of two subunits, large and small. The pair are made up of proteins and RNA molecules called ribosome RNAs. Congratulations, you have successfully made a long RNA molecule, but this isn't the end of transcription. The RNA has unrequired sections not needed to create proteins. This means we would need to remove these unneeded sections, otherwise known as introns. In other words, we'd have to go through DNA splicing, or else there would be far too many polypeptides. The needed sequences, however, are called exons.
DNA splicing is carried out by a signaling nucleotide called the snRNP, which are located at the cell nucleus. These snRNPs link together and with proteins to form a spliceosome. The spliceosome releases the introns and attaches exons together.
Hey, protein.
ASRP, what do you got there?
Oh, good stuff.
Oh, pass it over. We just made splicosome.
Lastly, the termination signal tells the polymerase releases itself from the DNA transcript.
Stop. You can release yourself right now.
Bet.
Now on translation at the ribosome sticks itself to the mRNA model to initiate the start codon, which is AUG, which signals the gene to be transcribed.
The tRNA molecules are made up of amino acids, and if you get the right amount of amino acids, you'll have enough to make a protein.
But he knows how to do at least like 3 months over. 3? Yeah. I can only do like as much. I'm sorry, that's just tough at home, right?
tRNAs are always finding their complementary strands, which are represented as triplets called codons. The start codon is AUG, and its anticodon would be UAC. UAC contains an amino acid called methionine, and then transfer it to the strand. The tRNA eventually leaves, but it will leave behind the amino acid it carried.
All these amino acids create a peptide bond together until it reaches a stop codon, which is UAG, and creates a polypeptide chain.
What's up, guys? So, now that we know each other, let's make some polypeptide chain. God.
In conclusion, the chain of amino acids were brought together based on the codes made by the mRNA, which was basically the assistant of the DNA. So in other words, the DNA was the director all along. And based on the type of protein structure it resulted in, folding and modification can vary.